Good evening to everybody and welcome at this inaugural lecture of the Institute of Neurology. So the inaugural lecture are a great opportunity for celebrating the new appointments and the great discovery done in both basic and clinical science at the Institute. It is also a great opportunity to celebrate exceptional citizenship, collaborative spirit, and excellence in training that are all quality that are all well represented in the two colleagues who will present their inaugural lecture tonight. I'm GP Schiavo, and I'm replacing for tonight the director of the Institute, Professor Mike Hanna, who cannot be with us tonight. And we start the inaugural lecture with Tammaryn Lashley. Tammaryn is a professor of neuroscience and is a former principal research fellow of Alzheimer's Research UK. Tammaryn undertook her PhD at the Institute under the supervision of Professor Tamas Revesz, who will give the vote of thanks at the end of her presentation. What is remarkable of Tammaryn is that she did her PhD maintaining a very important diagnostic histology role at the Queen Square Brain Bank from 1999. The research profile of Tammaryn grew with the help of two ARUK fellowships, both the junior and the senior fellowship. And this allowed Tammaryn to pursue her independent project in frontotemporal dementia. One of the big passion of Tammaryn is her commitment to training. And indeed, she became a departmental graduate tutor in the Department of Molecular Neuroscience and continued her role when moving to neurodegenerative disease. And this is very clear from the work that Tammaryn has done through the years. As I mentioned, the IUK has been very important for the training of Tammaryn and also of the other presenter. And Tammaryn, for her work, has been appointed chair of the UCL ARUK network. She was appointed the director of research at Queen Square Brain Bank to oversee the work using brain tissue in the many research projects running at UCL and beyond. And he has been elected the honorary secretary of the British Neuropathology Society. She has been awarded numerous grants from the MRC, Alzheimer's Society, the IUK, and the Michael J. Fox Foundation. And tonight we will hear her research and her achievement in and celebrate her professorship. Tammaryn.
Thank you, GP. I'll just share my screens with everybody. So thank you everybody for logging on this evening. It is unfortunate that we are not able to give our lectures in person this evening, but giving it over Zoom has meant that we are able to invite more people that wouldn't have made the journey to the Institute of Neurology this evening. So welcome everybody. I've entitled my talk, What We Are Not Seeing, as my research primarily focuses on seeing the pathological hallmarks of the different neurodegenerative diseases. Often being the first person, one of the first people to see these hallmarks down the microscope, but through many projects, I started to believe that we're not seeing everything when we look at the diseased brains. Before I can walk you through my journey that has culminated in my lecture this evening, I have some people that I need to thank that have made this journey possible. Firstly, I need to thank my home team. My husband, Andy, who is my rock, and without working as a team, none of this would be possible. My children, Eden and Ethan, who arrived during my PhD, and Erin during my postdoc years. They've all kept me grounded and focused on what matters in life. Thank you to my mum and dad for giving me the drive and determination never to give up. Mum, thanks for everything that you've done for me. You've always been there for whenever I need you. Unfortunately, my dad didn't get to see me start my PhD, but his attitude to life lives on in me. Thank you to my mother and father-in-law, David and Judy Lashley, who have both supported my career. Unfortunately, my mother-in-law passed away a few months ago, so although she was very proud of my promotion, she isn't here to listen to my lecture this evening. And finally, I have one huge thank you to Professor Revesz. This is when I'm glad I'm not actually giving the lecture in person because I can't actually see him giving me a raised eyebrow. But thank you, Tamas, for believing in me. I would certainly not be giving this lecture if it wasn't for you. I've learned so much about neuropathology, but I've also learned so much about geography, history, English grammar, and the list goes on. But most of all, I've learned integrity. I also have to take the opportunity to share with you where I began. Comments and corrections for one of my 70 figures in my PhD thesis, but through Thomas's continued guidance, I'm here giving you my professorial lecture today. So I started my journey at the National Institute of Medical Research in Mill Hill. I was there for 3 years working as a research technician before moving to the Institute of Neurology to work as a technician for Tamas, and I basically never left. I undertook my PhD, several postdoc positions, and through fellowship funding from Alzheimer's Research UK, was able to build my own research group. At the National Institute of Medical Research, I joined the team of the late Professor Jeffrey Reisman, initially supporting Professor Steve Davis's projects on spinal cord regeneration, and then supporting Xiaoli and Darching when Steve left for pastures new. This was where I learned the histology skills that are so important for my research today. From the EM techniques to the light microscopy, I got the buzz from seeing things for the first time down the microscope that nobody else had seen. And my first attempts at multiple stains to visualize some of the first olfactory and sheathing cell transplants in the corticospinal tracts. Whilst at the National Institute, as well as learning from many of the lab techniques that I use today, I learned so much more from the people that were there, including my science mums, Bernice and Yuandi, who showed me it was possible to have a work-life balance and were always there to give advice in any shape or form. Malvin was the go-to man when anything needed doing, ordering, fixing, or just about anything. And he also managed to get me involved in the Amateur Dramatic Society at the Institute, which I'm still not quite sure how he managed to do that. And Peter Woodhams for giving me all the time and teaching me what he knew in the lab. I also need to show you my lifelong friends, the microtome, the cryostat, and the vibratome. These are machines to cut the brain tissue into very thin sections. And without these machines, none of the work that I've done over the years would have been possible. And I actually feel like they are additional appendages to my body. I then moved to the Institute of Neurology in 1999 to start a one-year position as a research technician for Tamas. This one-year project was to investigate the expression of the tau protein in progressive supranuclear palsy, funded by the PSP Association. However, this project had not long started when I became involved in working on the familial cerebral amyloid angiopathy British type, a rare dementia where we could see using histological stains that there was an abnormal protein deposited, but at the time we didn't know what this underlying protein was. This also coincided with Tamas becoming the director of neuropathology at Queen Square Brain Bank. So time was taken up by running the diagnostic staining on the brains that had been donated to the brain bank. And when preparing these slides for this lecture, I came across old lab books with endless lists of days planned in the lab to maximize what I could get done in a day, even telling myself that I had an extra half an hour to cut more sections. In 2000, I registered for a part-time PhD with Tamas, Professor Janice Holton and Professor George Giso from New York University as my supervisors. One bright side to the COVID pandemic, that even after 20 years, I'm still having a meeting with all of them regularly on Zoom for a catch up, which is amazing to still get their advice on things when needed. For my PhD, I investigated the British type of dementia that we know today as the BRI2 gene-related dementias, which includes both the familial British dementia and the familial Danish dementia. I undertook the PhD part-time as I continued to work as a research technician to complete the diagnostic histology for the brain bank. Familial British and the familial Danish dementias are characterized by the deposition of amyloid. So I just wanted to stop and explain what the term amyloid means. It is used interchangeably with the A-beta protein that is found in Alzheimer's disease. Proteins such as A-beta, A-bri, and A-dan have certain unique sequences. However, these different peptide sequences can form different structures from the native protein to fibrils. It is these fibrils that we term amyloid, and we are able to stain the amyloid fibrils with certain dyes, for example, thioflavin. So when the British and dementia brains were first looked at under the microscope, the thioflavin stain showed the abnormalities, like we can see here with the image of a plaque from the British dementia, but the underlying peptide sequences were unknown. However, George and his team were able to identify the mutations associated with both the British and Danish dementias, with the British being a change in the genetic code that changed the normal stop codon, which meant the normal protein was extended by 12 amino acids, and the Danish mutation was a duplication insertion, which also meant the normal protein was extended. As the two proteins had extended parts, antibodies could be produced that could recognize these abnormal parts, and the proteins could be stained in the post-mortem brains. And this formed the basis of my PhD to characterize the pathology of both the British and Danish dementias. Using immunohistochemistry, we could see for the first time the A-bri plaques in the British dementia were found to be huge plaques in the hippocampus, as we can see here in panel A, and in the cerebellum, as we can see here in panel B, but also severe amyloid angiopathy, which is the protein found deposited within the blood vessels, and this was paired with severe tau pathology, similar to what we see in Alzheimer's disease. We also found the same in the British and Danish dementias, where we could see both 3 and 4 repeat tau in the neurofibrillary tangles. We also found that pairing the antibody staining with the thioflavin staining, that some of the deposits of the A-bri protein were found to be in pre-amyloid or non-fibrillar states. As we can see here, with the A-bri antibody, we can see an excess of the A-bri peptide compared to the amyloid stain of the thioflavin here in green. A similar study was carried out to investigate the distribution of the A-dan protein in the Danish dementia. And although the British and Danish peptides only differ by 12 amino acids, the underlying pathology was very different. There were more pre-amyloid deposits found in the Danish dementia, as we can see here with the A-dan staining compared to the thioflavin staining here. We can see a lot more red deposits than we can the green thioflavin. But one other distinguishing feature of the Danish dementia was the deposition of the A-beta peptide that's also found in Alzheimer's disease. As we can see here in the blood vessel containing the protein, we can see the A-dan peptide and then the A-beta peptide here. I also spent a lot of time using previous electron microscopy skills learned at the National Institute of Medical Research to determine that both the Danish peptide and the A-beta peptides were found in the same structure or the same pathological lesions. And here we can see that the A-dan peptide was stained with these large black gold deposits, and the smaller black gold deposits were sticking to the A-beta peptide. During my PhD, I also had the opportunity to go to New York University to learn how to biochemically extract and analyze the A-bri and A-dan from the post-mortem tissue. This was undertaken with my supervisor, Professor George Giso, and we had characterize the pathology of the brain, so now we needed to biochemically characterize the peptides. To do this, we use sequential fractionation methods to separate the soluble peptides from the detergent soluble and insoluble formic acid fractions. We showed that monomeric, dimeric, and trimeric AB species were present in all of the fractions, but in the insoluble fractions contained more of the AB peptide in aggregated states. Using mass spectrometry analysis from the isolated fractions, we were able to show that ABRI and ADAN also underwent post-translational modifications and formed pyroglutamate modifications in a similar manner to the A-beta found in Alzheimer's disease, meaning that these modified versions of the peptides were more toxic in the human brain. Further studies were carried out on the British and Danish dementias to investigate the presence of various molecular chaperones or amyloid-associated proteins to see whether they were deposited in the vascular or parenchymal amyloid lesions as previously seen in Alzheimer's disease. This included investigating the presence of ApoE, ApoJ, serum amyloid P, alpha-1 antichymotrypsin, cystatin C, and a range of heparin sulfate proteoglycans. We found that the deposition pattern of the molecular chaperones in the British and Danish dementias were similar to those found in Alzheimer's disease, suggesting they may also play a role in the amyloidogenesis or the formation of the amyloid fibrils. We also explored the mechanism behind the ABRI lesions using in situ hybridization to investigate the cellular origin of the precursor protein and the furin protein involved in the formation of the ABRI plaques. We found that the BRI2 messenger RNA and the BRI2 precursor protein were widely expressed in neurons, ganglial cells, and were deposited in the amyloid lesions in the British dementia. They were, however, not expressed in the cerebral vascular components, whereas the furin was found in a similar pattern, although it was expressed in the smooth muscle cells, suggesting that neurons and glial cells were the major source of the BRI2 precursor protein, and that the mutated precursor protein may undergo furin cleavage within the neurons to produce the amyloid peptide ABRI. We also investigated the systemic amyloid deposits, as we were in a position to study different organs from one of the British dementia cases. And this is a long-standing collaboration, which is still active today. And we have recently published a paper this year on the association of claustrin with both ABRI and ADAN peptides. After the submission of my PhD, I was fortunate to be able to continue working at the Institute of Neurology on several postdoctoral projects. These included observing Lewy bodies in the pathological hallmarks of Parkinson's disease, in the fetal grafts that had been transplanted into the brains of people with Parkinson's disease. Although the grafts were only around 10 years old, we found Lewy bodies within the cells that had been transplanted. This observation had key implications for the understanding and the pathogenesis of Parkinson's disease by providing evidence that the disease can propagate from host to graft cells. There was increasing evidence that A-beta may promote alpha-synuclein fibrillation. So in another project, we assessed the relationship between Lewy body pathology, A-beta deposition in 40 Parkinson's disease cases and 20 age-matched controls. In five cortical areas, we established the severity of the A-beta plaque load and the number of Lewy bodies. We found that the overall A-beta plaque burden, and in particular the diffuse plaque load, showed a significant correlation with the overall cortical Lewy body burden. The strength of this correlation was further increased in the Parkinson's disease cases with moderate to high A-beta plaque load, with the presence of the ApoE4 allele being overrepresented within this subgroup. And our data provided indirect evidence that the A-beta pathology is likely to be an important factor contributing to the cognitive decline of a subgroup of Parkinson's disease patients. Research was then directed towards frontotemporal dementia, where relating clinical symptoms to underlying pathology or disease mechanisms is a key challenge in the field, and particularly challenging to the heterogeneous disorders that make up the frontal lobe or degeneration spectrum. In collaboration with John Rohrer and others from the Dementia Research Centre, we re-evaluated 95 frontotemporal dementia cases that had been seen during life at the Dementia Research Centre, and then who'd gone on to donate their brains for research. This was a huge amount of work, as many of the brains had been donated prior to the current diagnostic criteria. So we needed to cut and stain all the brains again to be able to give them an up-to-date diagnosis. 51% of the cases had TDP43 pathology, 44% had tau pathology, and 5% had FUS pathology. And certain relatively specific clinico-pathological associations were identified. Semantic dementia was predominantly associated with TDP43 type C pathology, frontotemporal dementia and motor neuron disease with TDP43 type B pathology, and young onset behavioural variant FTD with FUS pathology. However, the most common clinical syndrome, behavioural variant FTD, was pathologically heterogeneous. This was a busy period with multiple projects being carried out, but I would always find little notes in my bag when I got to work, which made me organise things so I could get back home to the kids as quick as possible. Following on from the clinical path study, I was also fortunate to be able to detail the underlying pathology of a large cohort of FTLD FUS cases. These are one of the rarer subtypes of FTD, and we had 14 cases that had been donated or collected for research. First reports had been published detailing the FUS mutations had been found in ALS, and the FUS protein had been identified in the pathological inclusions. First reports from the rare FTLD cases showed that the presence of FUS in these inclusions, which prompted us to investigate the cases that we could not actually give a diagnosis to. So in collaboration with John Rohrer and Rena, we investigated the clinical neuropath and biochemical signatures of our cohort of cases. Neuropathologically, the cases had widespread FUS immunoreactive inclusions, as we can see here in the granular cell layer of the hippocampus. And we can also see the large motor neurons from the spinal cord contain pathological inclusions. These inclusions affected several major anatomical areas, including the cerebral cortex, subcortical, and brainstem nuclei. And we demonstrated biochemically that the FUS protein was more insoluble in disease than in the control cases. We also, for the first time, identified the presence of transportin in the pathological inclusions in FTLD FUS. Transportin is responsible for shuttling the proteins contained in the M9 nuclear localization signal between the nuclear and cytoplasmic compartments. RNA interacting proteins, including FUS, have been implicated as targets of transportin. The data from our immunohistochemical studies showed that FUS immunoreactive inclusions were also strongly labelled with the transportin antibody, and double immunofluorescence studies showed good co-localization between FUS and the transportin pathologies. These findings implicate FUS transport in the pathogenesis of FUS. It was at this point I began to think about potential fellowship projects that I could take forward independently, and the speed at which the frontotemporal dementia field was moving was exciting, with more genetic mutations were being identified and the pathological diagnosis changing with new proteins being identified. There was scope in the field of frontotemporal dementia, therefore, of adding knowledge to potential disease mechanisms in this large group of heterogeneous diseases. I was successful in obtaining an Alzheimer's Research UK Junior Fellowship to investigate the role of the nuclear transport system in frontotemporal dementia. I have to begin by thanking everyone at AR UK for their guidance and support for what you are doing, all doing to promote the need for more dementia research and funding within the area. There are so many of you that have supported and championed my research over the years. It truly feels like we do actually belong to part of a family. My focus was initially on the rarer FUS cases, where we investigated the presence of the HNRMP proteins in the pathological inclusions. This work was carried out by Priya, a research technician at the time, and we investigated whether HNRMP proteins were associated with FUS pathology. implementing dysfunctional nuclear export in the pathogenesis of FTLD FUS. HNRMP proteins were investigated in the affected brain regions using immunohistochemical analysis, biochemical analysis, and expression analysis. We demonstrated the presence of several HNRMP proteins in the pathological inclusions, including neuronal cytoplasmic inclusions, as we can see here with HNRMP A1, and also other HNRMPs that formed pathological inclusions or actually turned out to be FUS negative. The biochemical analysis revealed a shift of several HNRMPs from the nucleus to the cytoplasm, and the expression analysis revealed an increase in several HNRMP proteins in FTLD FUS, suggesting a wider dysregulation of the movement between the intracellular compartments than a mechanism only affecting nuclear import of FUS. FUS belongs to a family of proteins called the FET proteins. The other FET proteins, being EWS and TAF15, have also been found in a proportion of FUS inclusions. Other researchers have suggested that the FTLD FUS cases should now be termed FTLD FET due to the presence of the other FET proteins. However, we have shown the presence of transportin, and now with this present study, the presence of HNRMP R and Q in the FUS inclusions. Our identification of the frequent presence of HNRMP R and HNRMP Q in FTLD FUS inclusions suggests a potential role for these HNRMPs in the pathogenesis of FTLD FUS, and supports a wider role for the dysfunctional RNA metabolism rather than just centering around the FET proteins. This work was carried out as part of Lauren's PhD in collaboration with Professor Adrian Isaacs, with whom I've had many collaborative projects over the years. I presented my research at an ARUK conference in Manchester a few years back and coordinated these two studies with Professor David Mann, a neuropathologist from Manchester. We pulled our FTLD cohorts and found that HNRMP-E2 was associated with the pathological hallmarks that we observe in semantic dementia, or FTLD-TDP subtype C, and that HNRMP-A3 was found to be present in the pathological hallmarks in cases with a C9orf72 mutation. By identifying different proteins in the different subtypes of FTLD, we're able to build up a picture of pathways that could lead to the deposition of these proteins and learn more about the disease mechanisms. It was during this time that I also stained at different FTLD cases with the HNRMPK antibody. Looking down the microscope, I did not see that HNRMPK was found in any of the pathological inclusions. Instead, I saw an abnormal staining pattern that was different to what I'd previously seen with the other HNRMP proteins. We can see here what the staining looks like in the normal cases. We can see that the HNRMPK is found within the nucleus, and then lighter staining or weaker staining found within the cytoplasm. However, in the FTD cases, we found that the HNRMPK protein had moved from the nucleus, so we don't see any staining in the nucleus, and we can see these punctate staining of HNRMPK that's found within the cytoplasm. This wasn't specific to one FTLD subtype, but all of the FTLD cases showed this characteristic mislocalization of HNRMPK. This work was started by Sandrine and Yasmin and funded by Alzheimer's Society and Rose Trees Trust as an extension to my ARUK fellowship. We found that the HNRMPK was mislocalized in neurons that were found in layer 3 and layer 5 of the cortex, which corresponded to the pyramidal neurons within these two layers. And this was different to the set of neurons that contain the TDP43 or FUS inclusions that are used to diagnose the FTD cases. This work has now been taken forward by Alex, a PhD student in the lab, and Ariana, a postdoc within the lab, in collaboration with Jack, a UCL alumni who now is based in New York, Pietro, our resident RNA expert at the Institute of Neurology, and Emanuele Boratti from Trieste, who is a world-renowned expert for his work on TDP43. We expanded the pathological analysis to 61 disease cases and evaluated the HNRPK mislocalization in 35 controls of varying ages. In all cases, we found HNR-MPK mislocalization in the pyramidal neurons, which were mutually exclusive to the TDP43 and tau pathological inclusions. We found the mislocalization was prevalent in the FTLD cases and was also correlated with age in the control cases. We also showed that the mislocalization of HNR-MPK in neurons was not observed to co-localize with any mitochondrial, autophagosomal, or stress granule markers, as shown by double immunofluorescence. We see no overlap with the different markers. We know that HNR-MPK plays a major role in RNA processing within the nucleus. So if HNR-MPK is not found in the nucleus, the processes that it's involved in are going to be disrupted. Investigations carried out by Ariana showed that in cell lines where HNR-MPK had been knocked down, widespread changes were found in the transcriptome, which showed us the importance of having HNR-MPK within the nucleus. We found that without HNR-MP's regulatory function in the nucleus, then genetic information that should have been ignored was being included, and those that should have been included are being ignored, as we can see here by the inclusion of specific cryptic exons that Ariana found within the cell lines. From the 5 main cryptic events that she was able to identify, the inclusion of one cryptic exons was upregulated and found within the post-mortem tissue that we'd found the mislocalization previously with the immunohistochemical analysis. Future work utilizing single-cell approaches or alternative neuronal knockdown models of HNR-MPK will be important to identify cryptic events that are mechanistically relevant to the neurodegenerative disease phenotype, and investigate potential interactions between HNR-MPK and TDP43 within FTLD-related RNA misprocessing may be of mechanistic interest in itself. And this work is now being taken forward by Ariana, who's just been successful in obtaining her own brain fellowship. During my time reevaluating the FTLD cohort at QSBB, 2 cases stood out from the rest. It is unusual in the neurodegenerative field that we can identify a genetic abnormality by looking at the histological slides. However, one of these cases that have the C9orf expansion repeat, you can actually identify that the case has the expansion repeat by looking at the underlying pathology. It was whilst looking at over 100 cases, it became apparent that all the C9 expansion repeat cases had this particular pathology. In all cases, we found the TDP43 inclusions as highlighted here by the arrows, but we also see these dipeptide repeat inclusions in the C9orf cases. One case that we identified had a was homozygous for the C9orf expansion repeat and showed severe pathological features. In another case was reevaluated and the diagnosis brought up to date with the current diagnostic criteria. However, this case had already been known to have a progranulin mutation. To be thorough, I stained every case, regardless of the known genetic mutations, for the dipeptide repeat inclusions, and this case was also found to have the C9orf72 pathology. Genetic testing then confirmed the presence of the C9orf72 expansion repeat, an important finding that just because you have one genetic mutation, it doesn't mean that you won't have another one. It was now time to start thinking about my senior fellowship application, as I was over halfway through my junior fellowship. As work was continuing and expanding in relation to the HNR-MP proteins, I needed to think in which direction I could take my senior fellowship. Thinking back to the clinical path investigations and the finding that it was still difficult to determine the underlying pathology of cases diagnosed clinically with behavioural variant FTD, I thought this was an avenue that made sense to explore further. When cases are donated to Queen's Square Brain Bank, half of the brain is fixed in formalin and then coronally sliced from front to back. These 2 coronal slices are taken from around the middle of the half hemisphere of the brain, and it clearly shows the difference between a normal control and a case with frontotemporal dementia. we can see that the frontal cortex is severely shrunken or atrophied, and the hippocampus is also severely shrunken. The ventricles also become enlarged here and here. At the present time, it's difficult to predict the underlying disease mechanism from examining the patient in clinic or even examining the coronal brain slices from a case with behavioural variant FTD. Therefore, the gold standard practice of diagnosis remains the microscopic pathological analysis of the post-mortem brain. We know from the clinical pathological studies that someone with behavioural variant FTD can have underlying tau pathology, as we can see here from the different tauopathies. So somebody with behavioural variant FTD can have Alzheimer's disease with the presence of neurofibrillary tangles, have Pick's disease with the presence of Pick bodies, or cortical basal degeneration with the presence of astrocytic plaques. We also know if they have behavioural variant frontal temporal dementia, we can also find TDP43 pathology. Those with TDP43 pathology can be further divided into 4 major subtypes depending on the inclusions found. Type A having neuronal cytoplasmic inclusions, intranuclear inclusions, and short threads, primarily restricted to the second cortical layer. Type B can have cytoplasmic inclusions throughout all cortical layers. Type C, long twisted neurites, and type D, neuronal intranuclear inclusions. Or the patient with behavioural variant FTD can also have underlying phosphorylated. Therefore, it is of vital importance to be able to diagnose the correct disease mechanism during life for when disease-modifying therapies become available, so we can give the right treatment to the right people. For my senior fellowship, I utilised the cohort of FTLD brains at QSBB to try and identify biomarkers or hallmarks that could distinguish the different underlying pathologies, and several avenues of research were undertaken utilising the same brains, so that we could start to build up the picture of each of the brains donated and correlate all the different avenues investigated. One of these avenues was to undertake a head-to-head comparison of novel tau PET tracers that are being used clinically to diagnose tauopathies. This project has been running for many years and is a collaboration between myself, Kirsten and Eric at UCL Radiochemistry, Nick, John Schott and John Rohrer at the Dementia Research Centre, and Michael Scholl from the University of Gothenburg. Funded by my senior fellowship, Fidelity Bioscience and the Association of Frontal Temporal Dementias, PET is a promising tool to detect tau pathology in life, although limitations highlighted from the first generation of imaging agents sparked the development of novel tau PET tracers, which have entered clinical trials, but the details of the tau tracer binding at the molecular level need further investigations. Our collaboration aimed to evaluate the suitability of the first and second generation tau PET tracers for clinical applications such as differential diagnosis, patient stratification, staging, and treatment monitoring, both during the symptomatic and pre-symptomatic disease stages. We also use post-mortem tissue of a broad range of dementias, including those clinically that would have been diagnosed with behavioural variant FTD. To characterise the binding profile of cutting-edge tau tracers, we assess the tracer's robustness, sensitivity, and specificity to depict the tau. We have established collaborations with companies who have developed the leading tau tracers, and these companies provided us with their tracers to be able to test them as an independent academic partner. Our first 2 studies characterise first-generation tau tracers in Alzheimer's disease, primary tauopathies, and other dementias, including those with TDP43 pathologies. We assessed the binding of AV1451, also known as fluotausapia, in a range of dementias. Phosphoimaging was used to quantify binding in the post-mortem brain from 33 cases, where fluotausapia showed high specific binding in cases with Alzheimer's disease, moderate binding in Pick's disease and frontal temporal dementia with Parkinsonism, but low displaceable binding in corticobasal degeneration, supranuclear palsy, and non-tau proteinopathies. In a second study, work was carried out by a PhD student, Melissa Wren, who compared various first-generation tau tracers, including for tau superior and the THK series of tracers. As all of the tracers were now being used in clinical research, there were concerns about off-target binding and low sensitivity of these tracers. We only observed tau tracer binding in the cases with Alzheimer's disease and one case with frontotemporal dementia with the R406W MAPT mutation that also contains both 3-repeat and 4-repeat tau, as is seen in Alzheimer's disease. Our results highlighted limitations of the first-generation tau PET tracers, in particular the lack of correlation between pathological tau, tau tracer binding, limited sensitivity to tau in early disease, and high variability of tracer binding between and within cases. Whilst the new tracers were now entering clinical studies, there was limited information available to assess their suitability for clinical applications. Therefore, we undertook more head-to-head comparisons, which were urgently needed to understand the differences in the radio tracers' binding profiles. This study was undertaken by Barbara, a postdoc in my lab, and Stephen Yap, who is a postdoc in radiochemistry. We characterized the binding of 4 second-generation tau tracers in the post-mortem human tissue from a cohort of 25 dementia cases and age-matched control using quantitative phosphoimaging. 4 radio tracers depicted tau inclusions composed of paired helical filaments with high specificity, both in cases with Alzheimer's disease and in primary tauopathy cases with additional Alzheimer's disease pathology. In contrast, cortical binding to primary tauopathy cases without paired helical filaments was found to be within the same range as the normal age-matched controls. The high variability of cortical tracer binding within the Alzheimer's disease group followed the same pattern with each tracer, suggesting that all compounds were suited to differentiate Alzheimer's disease from the other dementias. So moving forward, we are still in need of tracers to distinguish the 2 different types of isoforms of tau, the 3-repeat and 4-repeat tau, and for a tracer that would be able to detect the underlying TDP43 pathology. We are now also moving forward with an exciting project that's also funded by the Association of Frontotemporal Dementia to investigate neuroinflammation tracers that might have a potential to stratify patients with frontotemporal dementia. We will explore the suitability of the next generation neuroinflammation PET tracers that highlight activated microglia as a biomarker for all early disease diagnosis and disease progression in FTD. Tissue from the same brains used for the tau PET tracer studies were also used for in a study investigating the neuroinflammation in FTLD. This project was undertaken by a PhD student, Ione, who examined regional patterns of microglia burden, activation, and dystrophy in sporadic and genetic FTLD. Immunohistochemistry was performed in frontal and temporal cortex from 50 pathologically confirmed FTLD cases, using markers to detect phagocytic, which are the CD68 microglia stains here, antigen presenting, which is the CR343 microglia stains here, and the resident microglia are the IBA1 positive cells here. Microglia burden and activation, or the morphology of the microglia, was assessed quantitatively for each microglia phenotype. IBA1 positive microglia were assessed semi-quantitatively for dystrophy severity and qualitatively for rod-shaped or hypertrophic morphology. Microglia were compared with each region between the FTLD cases, AD cases and controls, and between different pathological subtypes of FTLD. We found a higher burden of phagocytic and antigen-presenting microglia in the FTLD cases and AD cases compared to the controls, but the activation was often not increased. The burden was generally higher in the white matter compared to the grey matter, but activation was greater in the grey matter. The results from this project will also be important to evaluate against those that are generated from the neuroinflammation tracer study. As the accumulation of abnormally phosphorylated TDP43 is one of the main pathologies affected neurons of people with frontotemporal lobe degeneration, and we diagnose and classify cases on the morphological diversity and the neuroanatomical distribution of the TDP43 accumulations. As with other neurodegenerative diseases, it is important to understand the molecular basis of the proteins found within the pathological inclusions. As FTLD TDP is subdivided into 4 major subtypes, understanding the molecular basis of the protein in the 4 subtypes may give us insights into the underlying mechanism of deposition. As you can see from this TDP43 immunohistochemical preparation, we have neurons with normal TDP43 staining, where we can see the TDP43 staining within the nucleus. And we also have neurons that are harboring a pathological inclusion. We can see here where we lose the nuclear TDP43 staining, and the TDP43 protein accumulates within the cytoplasm. So the difficulty in trying to extract and analyze the TDP43 protein is that we also have to extract the normal TDP43 protein along with the pathological protein. So in collaboration with Magda Polymenidou from Zurich, we used the same cohort of FTLD cases to develop a sarcospin protocol, a new method for biochemical isolation of pathological TDP43, which we believe will be a valuable tool to extract the pathological TDP43 from these cases from the physiological protein itself. Using the sarcospin extraction method on the different FTLD TDP subtypes, we showed that the phospho-TDP43 extracted from the brains form stable assemblies of distinct densities and morphologies that are associated with the disease subtypes. Pairing the biochemical extractions with mass spectrometry, we also revealed proteins beyond TDP43 that become abnormally insoluble in a disease-specific subtype manner. The proteins identified by mass spectrometry were not proteins that were found in the pathological inclusions, but were often found in other cell types like FBOX2 in astrocytes in the FTLD TDP-A subtype, suggesting that they probably represent a molecular signature originating from the divergent pathological mechanism characterizing the subtype. Most critically, the biochemical and morphological differences between the TDP-A and C subtypes, or the TDP43 assemblies that were extracted from the different subtypes, were associated with differential seeding and neurotoxicity potential. Indeed, the TDP-A aggregates extracted from the subjects from fast progressing disease caused toxicity and induced endogenous TDP43 aggregation in the cellular models, whereas the TDP-C aggregates from slow progressing disease seemed inert under the same conditions. Additional work is necessary to establish a direct link between TDP43 pathological characteristics and disease subtypes, but this study suggests that disease heterogeneity could originate from alternate pathologically TDP43 conformations, as has been observed for the case of prion strains, and opens up avenues for creating cellular and animal models. Several projects and collaborations have also been maintained or developed over the years whilst undertaking my junior and senior fellowships that have been focused on frontotemporal dementia, primarily because the subject of these projects are so interesting that they just needed to be needed to carry on. One of these studies was carried out by Christina during her PhD, which was funded by Alzheimer's Research UK, and she investigated an area in the hippocampus called the Presubiculum. And we can clearly see the Presubiculum, the area highlighted on the A-beta immunohistochemical preparation, as a large area that we nicknamed in the lab called the smudge. We had seen this many years ago, that this area of the hippocampus had a different pattern of A-beta deposition compared to the rest of the hippocampus. We also found the same pattern in the British and Danish dementia. In fact, the same phenomenon is seen in any disease that has extracellular amyloid deposition. So in this study, we set out to compare the presubiculum with the entorhinal cortex, an area that has classical A-beta plaques. And we investigated the presence of neurofibril tangles, the level of tau pathology, and found that the presubiculum, there was overall reduction in tau pathology compared to the entorhinal cortex, and less neurofibril tangles was observed in the presubiculum, again compared to the entorhinal cortex, in both sporadic and familial Alzheimer's disease. We then investigated the level of microglia activation in the presubiculum compared to the entorhinal cortex. And then, although we could see no difference when staining with IBA1, which stains for the resident microglia, when using stains for CD68 or CR343 to show the activated microglia, we only found activated microglia in the entorhinal cortex, which contains the classical A-beta plaques, and no activated microglia in the presubiculum, which contains the smudge area of the A-beta. We utilised laser capture microdissection to be able to cut out the presubiculum here and the entorhinal cortex to investigate the A-beta peptides that were present in both areas. Unsurprisingly, we found truncated A-beta species and post-translationally modified A-beta species within the entorhinal cortex, but the majority of the A-beta found in the presubiculum was a full length or truncated at position 4. We also undertook pathway analysis of the proteomic data from both regions to identify potential tissue factor differences that could predetermine the fate of the extracellular A-beta. We identified a downregulation in the annexins in the presubiculum and further worked to investigate the whole proteome data from the laser captured samples may yield tissue differences between the two regions. And we just started a collaboration with Afia Ali from the School of Pharmacy to look at the functionality of the neurons in both the presubiculum and the entorhinal cortex. Staying with the theme of A-beta deposition, Nanette Williamson, a PhD student who was also funded by Alzheimer's Research UK, investigated the variability and layer distribution of A-beta in familial Alzheimer's disease. So she took the brain tissue from 20 familial Alzheimer's cases and cut sequential sections from the frontal cortex and stained for A-beta and Nissl stain to be able to define the cortical layers. The frequency of the different A-beta plaque types were graded for each cortical layer, and the severity of the cerebral amyloid angiopathy was determined in the cortical layers and the leptomeningeal blood vessels. Comparisons were made between the familial Alzheimer's disease mutations and the ApoE4 status, with associations between pathology, clinical, and genetic data was investigated. In this cohort, the possession of the ApoE4 allele was associated with increased disease duration, but wasn't associated with age of onset. We found A-beta pathology to be heterogeneous between cases, although the A-beta load was the highest in the cortical layer 3 for all mutation groups, and higher A-beta load was associated with the ApoE4 allele. Cotton wool plaque frequency was positively associated with the severity of cerebral amyloid angiopathy in the whole cohort, and carriers with the same PS1 mutation, as we can see here, had differing patterns of A-beta deposition across the cortical layers, potentially because of different risk factors. So this one carries an ApoE4 allele, and this one carried 2 ApoE3 alleles. In another long-standing and fruitful collaboration with Joe and Natalie, we are investigating the pathological determinants of white matter hyperintensities. So white matter hyperintensities are frequently seen on brain imaging in both familial and sporadic Alzheimer's disease. And these lesions can also be seen in the aging brain and with those with vascular risk factors. White matter hyperintensities are usually considered to be a marker of cerebrovascular disease. However, they are also seen in some young onset Alzheimer's disease patients who lack the vascular risk factors. And actually attributing the clinical significance to these imaging features is extremely difficult. In one study, we investigated the white matter hyperintensities in 52 familial Alzheimer's disease patients to see if there was any associations with the mutation position, and 10 of the mutation carriers actually donated their brains for research. We found increased white matter hyperintensity was observed in the PS1 mutation cases where the mutations were after codon 200. The white matter hyperintensity burden on the MRI correlated with the severity of CAA and the presence of the cotton wool plaques in several areas. This has now been extended to investigate the pathological correlates of white matter hyperintensities in both sporadic and familial Alzheimer's disease. And we've now set up a pipeline to be able to investigate this in prospective brains that will be donated to Queen Square Brain Bank. MRI scans that are carried out during life often are undertaken many years before the brain is donated. Therefore, having an up-to-date and accurate picture of the white matter hyperintensities is quite rare if the scans are taken years before the brain donation. In this pipeline, Phoebe, a PhD student who's supervised by myself and Dr. Joe Barnes, set up this pipeline to enable us to scan the brains that have been donated and obtain a post-mortem MRI. This will enable us to visualize the white matter hyperintensities that are present in the brain tissue and locate them to undertake pathological analysis to identify any biological or pathological correlates. In-life scans will be compared to the post-mortem scans, and the brain will undergo detailed macroscopic dissection where photographs from each coronal block will be taken so that we'll be able to register the exact brain regions back to the MRI scans. This is made possible by the help of the neuropathologists at Queen Square Brain Bank, previously Jan and now Zane, who help with the blocking of the brains used in these studies. Sequential sections are then cut from the blocks of interest that contain the white matter hyperintensities, and histological evaluation will be undertaken. Phoebe was able to set up the pipeline during her PhD and look at selected markers for myelin, axons, glial cells, and vascular integrity. And now we're just waiting more brains that fit the criteria to be donated to take this project forward. So I'd like to thank my previous team members, postdocs Sandrine and Yasmin, who have both expanded their families and spending time with them. Research technicians, Priya, Bridget and Tanisha, who have all been more than technicians and are involved in so many projects. Priya has now undertaken her PhD at the Amsterdam Brain Bank, and Bridget her PhD at the University of Sheffield. I'd also like to thank all the PhD students that I've supervised, and I've learned so much, or they've taught me so much over the years. Thank you to my previous heads of departments, Nick and John as head of molecular neuroscience, and Nick and Sarah as head of neurodegenerative disease. You've all been generous with your time and offered so much needed guidance when needed. Professor Blas Frangioni for hosting me in New York during my PhD, and for the numerous chats I've took every word on board. Linda for being an amazing mentor and give me a push when I needed to apply for my senior fellowship. Martin for his much needed guidance over the years. Hugh and Henry, who both undertaken their PhDs when I started mine. Thanks for giving me so much advice over many years. The late Professor Robert Balash for his lunchtime chats, advice, guidance, and interesting papers that he would always forward me to read. Andrew and Tom for having me and my team based at Queen Square Brain Bank for so many years. Henrik for being Henrik. Apologies, Henrik, I've not been able to include any projects that are being undertaken with yourself or your team in Gothenburg. We have met so many collaborative projects that are ongoing. Rena and Rowan, who I feel like I've grown up with, have always been there when I've needed them in whatever capacity. And one, oh, it won't move on. Oops, sorry. And finally, this is my last slide, I promise. I'd like to thank Agada and Marta for all their support over the years, and apologies, Marta, that Thomas keeps writing more papers. To Kate, Hill, Rob and Steve, who again have been there for me from the beginning and seen, how hard it got sometimes, but I got there in the end. All QSBB staff, past and present, Anne, Susan, Linda, Karen and Milan, and now Illy and Lynn, who are all amazing. Thank you for all your support over the years. Previous postdocs, Shani and Aoife, Zainab, who is a current PhD student working on posterior cortical atrophy. Apologies, Zainab, that I haven't had time to talk about your project. So he's the font of all knowledge with regards to epigenetics, so watch this space because great things are coming. Shannon, who I met while she was collaborating on one of her PhD projects with Professor Reel at UCL. And finally to Rosie and Imran. Imran in molecular neuroscience and Rosie in DND. Thank you for putting up with all my constant emails regarding grant applications and pretty much anything else. I really don't know how you both do your jobs, and we would all be lost without you. Thank you.
Thank you, Tammaryn. I would like now to invite Professor Tamas Revesz to give the vote of thanks.
I feel very privileged that on behalf of everybody, I have the opportunity to thank Tammaryn for this wonderfully executed lecture. I have no doubt that everybody agrees with me that this lecture truly reflected Tammaryn's experience and excellence in many areas of her chosen field of neurodegenerative diseases. It was particularly satisfying for me to be asked to thank Tammaryn today, as I could witness her remarkable progress throughout the years. I met, as Tammaryn also mentioned this, I met Tammaryn in 1999, in January 1999, when I was among the first whose project was supported by the PSP Association. We had several applicants for the research technician position, which was funded by the grant, but Tam's CV offered the skills we were looking for. And her performance at the interview made it absolutely clear that we wanted her to work on our project. Tammaryn enlisted the projects which followed, but I would like to mention a little bit more her part-time PhD project, in which, as she mentioned, she worked on familiar British dementia and familiar Danish dementia, which were then newly discovered exciting diseases. She also went to New York, working under the wings of George Giesel. I would like to mention her viva, where Tammaryn impressed her examiners by the quality of her thesis and also her knowledge of the topic. And as a consequence, I think her PhD viva was one of the shortest, which actually was quite disappointing for her. She would have wanted to spend more time with her examiners and to discuss and entertain them with further details of her work. We heard that with the help of her considerable grant income, Tammaryn established her own research projects and research group. Now, as director of research at the Queen's Square Brain Bank, she advises and helps several young researchers with their projects. Tammaryn has become a well-liked and respected teacher, supervisor of many PhD and MSc projects. Having known Tammaryn for over 22 years, I, as a colleague, if I would need, as a colleague, to characterize her concisely, among the many words, I would certainly use the following: motivation, curiosity for identifying unsolved questions, dedication to work, consistency, and also very importantly to others, including me, helpfulness. Tammaryn, on behalf of everybody, let me conclude with thanking you again for this excellent lecture and heartfelt, warmest congratulations. Thank you.
Thank you, Thomas.
Thank you, Tamas, for this beautiful vote. Tammaryn, you have the chat is completely full with comment, and I apologize that this is a plenary lecture, so no questions are allowed. So thank you. Thank you, Tammaryn, and congratulations.